Hello everyone, welcome to Ibdp Biology. Today our topic is going to be about ultrastructure of cells. Today we're going to discuss the prokaryotic and eukaryotic cells as well as electron microscope and how we see the organelles of prokaryotic and eukaryotic cells under the microscope. Can we identify them or not? So let's get started. First of all, we'll start with the prokaryotic cells. Maybe you've heard the name prokaryotic, but you don't truly know what it is. Prokaryotes are single-celled organisms that are the earliest and most primitive forms of the life on Earth. And prokaryotes include bacteria and archaeans. Some prokaryotes, such as cyanobacteria, are photosynthetic organisms that are capable of photosynthesis. The first photosynthetic organisms are the cyanobacteria. Prokaryotic bacteria can be found almost anywhere and are part of the human microbiota. They live on your skin, in your body, and everyday objects you can see in your environment. So prokaryotes have a simple cell structure without compartmentalization, and it means that they are prokaryotes. Pro, it means before the nucleus, so it means that they do not have the nucleus and certain organelles. And this is the scanning electron micrograph of Escherichia coli, a common type of bacteria. This is the size of the 1 micrometer. So prokaryotic cells have the cell wall, pili, plasma membrane, flagellus, ribosome, nucleoid region containing DNA. So let's find out how they are working. So prokaryotic structure and functions. First of all, let's get to know the cell wall. That cell wall protects and maintains the shape, composed of peptidoglycan, proteins. Some have additional layer to adhere to the structure, such as teeth and skin. Plasma membrane controls movement of materials in and out, role in binary fission. Cytoplasm is the complete interior, no compartments, and DNA most visible structure here. What about pili? These hollow hair-like structures made of protein allow the bacteria to attach, like look at this, those pili, attach to other cells, and specialized pili have a role in conjugation. Flagella, this is the long whip-like thing is the flagella, and they are actually cytoplasmic membrane. Bacteria may have one or few or many flagellas in different positions on the cell. Ribosome is a site of protein synthesis, small, made up of two parts that come together and can be in large numbers. This is in prokaryotic cells, size of the ribosome is 70S. So S is a Swedberg unit, a measure of the rate of sedimentation and centrifugation rather than size, and accounts for why fragment names do not add up this one. So nucleoid region, DNA in the bacterial cell is generally confined to the central region, and though it isn't bound by a membrane, it is visibly distinct from the rest of the interior, as you see here. Chromosome and the plasmids are here, as you can see on the microscopic picture. The chromosomes and the plasmids are not visible, but chromosomes you can see here. Plasmid is a DNA molecule that is separate from and can replicate independently of the chromosomal DNA, and they're double-stranded and in many cases circular. Plasmids usually occur naturally in vector, but are sometimes found in eukaryotic organisms as well. So a drawing of the ultrastructure of prokaryotic cells based on electron micrograph. Which structure can you identify in this electron micrograph? So this is a typical rod-shaped bacteria, and this is Bacillus coagulans, which under the microscope we took a picture. So find out the similarities and the same parts here, so you can draw them and write down. You must be able to draw and label a diagram of the ultrastructure of a prokaryotic cell based on electron micrograph, and the organelles previously discussed must be included in your drawing, and only draw and label features you can clearly see. Don't put structures in because you think they should be there, such as flagella. We don't see the flagella here, so do not put flagella there just because you think that they should be there. Prokaryotes reproduce asexually using process of binary fission. The DNA is replicated in the two DNA loops attached to the membrane. The membrane elongates and pinches off. The cytokinesis happens in two separate cells. Two daughter cells are genetically identically produced here, as you see. Here, the plasmids and DNA is actually replicated, and then the cell starts to divide to two daughter same cells here. Similarities between prokaryotes and eukaryotes is the DNA, the plasma membrane, cytoplasm, and ribosomes. These are like actually four simple organelles that prokaryotes and eukaryote cells have together. Differences, in prokaryotic cells, DNA is just a ring, but there is no protein. But in eukaryotes, we've got the chromosomes, and it's inside the nucleus. In prokaryotes, DNA free, but in eukaryotes, it's inside the nucleus. And there is no mitochondria in prokaryotes, while mitochondria exists in eukaryotic cells. There is a 7S ribosome, but in eukaryote there is 80S ribosome. In prokaryotes there is no internal compartments, but in eukaryotes there are organelles with their own membrane. Prokaryotes are less than 10 micrometers, but eukaryotes are greater than 10 micrometer. So eukaryotes have a compartmentalized structure. What does it mean, a compartmentalized? So eukaryotic cells are divided into compartments such as organelles by partitions known as membranes. They have many different compartments which are separated by double or single membranes such as the nucleus, mitochondria, and ER. So you can read it on page 21 in your text and summarize the advantages of being compartmentalized. And like here we have seen the advantages of being compartmentalized: efficiency of metabolism, localized conditions, toxic damaging substance can be isolated. So and numbers and location of organelles can be changed depending on cells' requirements. Eukaryotic cells and idealized animal cells and idealized plant cells. Let's see what we have here. This shows the organelle of a liver cell, and we can see the organelles here: the plasma membrane, RER, mitochondrium, lysosome, Golgi apparatus, nucleus. Eukaryotic cells are usually 5 to 100 micrometers. Nucleus is membrane bound and have membrane bound organelles, such as a nucleus. Generally, it's spherical with double membrane pores. It has the pores in the membrane because it produces ribosome and contains genetic information and chromosomes. So the 2 meters of human DNA fits into a nucleus that is like millions, 0.5 meters across. The mitochondrion, think of the mitochondrion as the power of the cells. We know from the previous year that we learned the aerobic respiration that mitochondria takes an important role in the respiration. It has a double membrane, smooth outer and folded inner. Folds are called cristae. A shape varies, site of energy production during cellular respiration. They have their own DNA, a large surface for cellular metabolism, and contains its own ribosomes and can produce independently. Free ribosomes are the 80S ribosome, larger than the ribosome found in prokaryotes. No membrane. These appear as dark granules in the cytoplasm, synthesize proteins to function in the cytoplasm, and bound ribosomes are attached to an outer organ called the endoplasmic reticulum, and the protein products are shipped out of the cell. So you see these free ribosomes are in the cytoplasm and bound ribosomes next to the rough endoplasmic reticulum. And the plasmic reticulum are divided into two parts: smooth ER and rough ER. The smooth ER has no ribosome It synthesizes the pancreatic cells, synthesize lipids, and rough ER consists of the flattened membrane sacs called cisternae, often located near the nucleus. ADS ribosome attached to the outside, synthesize protein which are transported by vesicle to the Golgi apparatus before secretion out of the cell. And pancreatic exocrine cells would have an abundance of these due to constant production of digestive enzymes. Golgi apparatus, as you see here, the Golgi apparatus, and here you see the Golgi apparatus is a cytoplasm of macrofauna lung tissue. Like the ER is composed of the flattened sacs, call it cisternae. However, it has no ribosome and is often near the plasma membrane. Cisternae are shorter and more curved than those on ER. And the Golgi collects, packages, modifies, and distributes material the cell makes, especially the proteins. So as you see, in the rough endoplasmic reticulum, those attached ribosome produce protein, and those proteins by vesicles get into the by cisterns go to the Golgi apparatus, and Golgi apparatus distributes these proteins to different part of the cell, even to other cells as well. Vesicles, a single membrane with fluid inside and very small, used to transport materials outside of the cell. The lysosome, intracellular part of the cells arise from Golgi, because Golgi apparatus produces lysosome. It's a spherical with a single membrane. Now, stains dark in micrograph due to high concentration of enzymes. Lysosome carry inside the lysosome. It has the acetic enzymes, because lysosome also ingest food, break down food. Also, we can use it in break down damaged and unwanted organelles, and cell suicide when one of the cells are already lost its function. The lysosomes comes and engulf it, so the cells are actually getting rid of the old unwanted cells as well by lysosomes. So here I want to show the lysosome here. We're gonna watch a video now with you. So let's see the video about lysosomes.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles such as old mitochondria. This results in the destruction and recycling of these structures.
You can see the lysosome here, and as you know, the lysosome is actually the role of the lysosome is breaking down the food, breaking down the viruses, bacteria, and as well as the cell suicide. Here, vacuoles. Vacuoles are the single membrane with fluid inside. In plants, cells vacuoles are larger and permanent, often occupying the majority of the cell volume. But in animals, vacuoles are smaller and temporary and used for various reasons, such as absorbing food or digesting it. Store food, waste, toxins, water, increase surface area to volume ratio, and allow plants to be rigid, because they need to be rigid, and they need to be in a state of turgid to go against the wilting. Cilia and flagella. Cilia, tiny hair-like projections, contain microtubules used to move the cell or move to fluids next to the cell. such as lung cilium, or the we can see here in uterine tubes. Also, we have the cilia's flagellas. There are thin projection from cell surface, contains microtubules used for the movement. Mature plant cells do not contain this structure, but some plant gametes do. Microtubules and centrioles. Microtubules, small cylindrical fibers called microtubules, have a variety of functions, such as part of the structure of flagella, and they play a role in cell division. Centrioles consist of two groups of 9 triple microtubules and are mainly found in animal cells, not present in vascular plants or fungus. So here the chloroplast in plants but not animals contain double membrane, same size as bacteria, has its own DNA, has 70S ribosome, can reproduce on own. Inside are the stacks of thylakoids and usually oval in shape, site of the photosynthesis. Starch granules may be present if producing lots of food. And think of the chloroplast as the solar panel of the plant cell. Only plants and algae have chloroplast. Granum flattened sacs like solar panels, and stroma fluid contains enzyme and cold chemicals. There's thylakoids and stromers are the actually increasing the surface area for the photosynthetic process. Cell wall, usually we see in plants, not in animals. Extracellular component, not an organelle, secreted by all plant cells as well as the fungus. In plants, it contains mostly cellulose. Cellulose is permeable, strong, and hard to digest, but in fungus, it's actually, we call it a cuticle. Peroxisomes, similar to lysosomes, except peroxisomes bud from the endoplasmic reticulum, not from the Golgi, have many roles, involvement in producing bile, fatty acid, breakdown of the cholesterol, myelin production, so getting rid of the peroxide, so etc. Extracellular matrix in animal cells composed of collagen and glycoproteins form fibers anchored to the plasma membrane to strengthen it. Cell-to-cell interaction happens by extracellular matrix, leads to coordination of cell action, may be involved in directing stem cells, and possibly cell migration and movement also take place by the extracellular matrix. Cytoskeleton. The name is misleading. The cytoskeleton is the skeleton or scaffolding of the cell, but it's also like the muscular system as well, able to change the shape of cells in a flash, such as we can say the lysosomes or phagocytic processes goes on by the cytoskeleton. A macrophage using the cytoskeleton to reach out for the hapless bacterium, or membrane ruffles on a crawling cell. So centrosomes, usually a pair of centrioles, assemble microtubules for structure and movement. Involved in cell division, higher plants produce microtubules even though they do not have centrioles. All right. So differences between plant and animal cells. Plants has the cell wall, chloroplast, but animals do not have this one. Plants has the large vacuoles, but animals have the small or non and temporary. Plants store the carbs as a starch, animal as a glycogen. Plant has no centrosome and it's a fixed angular shape, but animal has a centrosome and its shape is a flex. So we've got the review part, define prokaryotic cells, and discuss the difference between eukaryotic and prokaryotic cells. Talk about the organelles of the eukaryotic cells, what is the function of the plasmid, what is binary fission. So here we have the electron microscope, and we're going to talk about the electron microscope, which has a much higher resolution. And resolution is defined as the shortest distance between 2 points that can be distinguished. So light microscopes has a limited resolution, but electrons have a much shorter wavelengths. Therefore, electron microscope have much higher resolutions here. Light microscopes are usually limited to 1000 because due to resolution, nothing is gained by increasing the magnification. Try zooming in on an image on your laptop or phone after a certain point. That's why resolution is very important. So here, the ultrastructure is all the structure of biological specimen that are least 0.1 nanometer in their smallest dimension. So light microscopes allow us to see the structure of the cells, but electron microscopes allow us to see the ultrastructure of the cells. An electron microscope can see viruses 0.1 micrometer in diameter, but light microscopes cannot. So in electron micrographs, so we can see a rat liver cell with color enhancements to show organelles. Exocrine gland cells of the pancreas, the structure of function of organelle within exocrine gland cells of the pancreas, you can see here, and find out the plasma membrane, mitochondria, nucleus, rough endoplasmic reticulum, Golgi body, vesicles, and lysosomes. In palisade mesophyll cells, let's find out the cell wall, plasma membrane, a chloroplast, mitochondria, vacuoles, and a nucleus here. Okay, so here we just looked at the one pancreas cells and palisade mesophyll cells. Now draw a pancreas exocrine cell form, the image below. You should be able to label the following structures such as the plasma membrane, mitochondria, RER, nucleus, and secretory granules, which is the dark spheres. Here, the cell wall, vacuole, and the chloroplast we can see of the eukaryotic cells and on an electron micrograph here. So which structure can you identify in this false colored TEM image? So liver, it's a liver cell, so let's find out what we can see. So here we can see the plasma membrane, a mitochondria, the cytoplasm, free ribosomes. There are a lot of free ribosomes in the liver cell. In the nuclear membrane, nucleolus, lysosomes, which is yellow, rough endoplasmic reticulum, and Golgi apparatus here. What organelles can you identify here? Think about the role of the organelles that occurs most common and deduce the function of the cells. Here we can see that nucleus is present, no cell wall, RER is present, lots of mitochondria, probably the synthesis of protein requires energy, lots of secretory granules, and likely to be a cell that specializes in secreting a protein product. And it is in fact the mammalian exocrine secretory cell from the pancreas. So what organelle can you identify in the topmost layer of the cells? Let's find out. We see the nucleus is present, no cell wall, cells closely packed, and has a cilia dominated and likely to be a protective layer. And it's in fact a ciliated epithelial cell from a mammalian lung here. What about this picture? What organelle can you identify here? We can see that a cell wall is present, no chloroplast, vacuoles are relatively small, and nucleus are relatively large, and possibly recently divided cell tissue from a plant root, and is in fact it's a plant cell found at the root tip. So guys, thanks for watching. Have a nice day. Bye bye.